Good morning, viewers. I am Professor S. Jeevan Chandra from Chaitanya Deemed University. I would like to explain about cell signaling mechanism, a way of molecular language of cell communication. Many prokaryotic and eukaryotic cells they are interacting with each other in a chemical manner. An information is called a cellular signal is intended to carry out a biological function. The signals are vary in biological nature. They may be simple chemicals, amino acids, proteins, or their derivatives. They are effectively diffused from external environment into biological interior and they carry different functions. The way of communicating with each other is called cellular communication, and the process of cell communication in a cascade manner is called cellular signal transduction. The cell signaling will be of four ways. One is autocrine mechanism. The signal molecule will arise from the same cell and received on the same type of receptor from single cell origin called autocrine mechanism. The signals which are produced from the cell and they received by neighbor cell or distant cell called paracrine signaling mechanism. And some of the signals produced by a particular gland and they entered into circulatory system. Through a circulatory system the distant cells can be responded, that is endocrine type of systems. And contact based gestokine based on the sensing and pressure or other physical attachment and physical touch can be deliver a signal called juxtapositioning and contact based signaling mechanisms. There is a purpose for signal transduction in cellular process. A cell has to receive the signals for its survivability, or it could be the process of division of cell into two different types, or differentiation of the cell from one another, or it may be converted into another type of cell. For example, stem cell will be differentiated into different kinds of cells. So finally, in some occasions the cell signal may be suicide or death of a particular infected cell also there. Most of the endocrine signaling mechanism will leads to response of long distance cells via circulatory system or a blood system. Example, parathyroid hormone or adrenal hormone or hormonal systems. Paracrine system is a local type of system where neighbor cells surrounded by a cell can be responded to the stimuli via diffusion of the signal molecule. Example, the vascular endothelial cells, granulocyte, insulin like growth factors, and other systems. Autocrine mechanism will be received in the same cell from the same origin. Example, for insulin like growth factor, which is going to control the cell growth and cell mass. The diversity of signal response molecules. The diversity of response molecule can be different types. The receptors can be G protein linked receptors, enzyme linked receptors, and ion channel receptors, steroid hormonal receptors. Coming to class of ligands, ligands could be of two major groups: hydrophilic ligands, hydrophobic ligands. Hydrophilic ligands will dissolve in water and blood, they can be easily diffused. But a hydrophobic molecule could be able to cross the membrane and reach the interior of nuclei, and within the nucleus particular gene can be controlled. So hydrophobic receptors are very important. Most of the hydrophobic receptors are steroid hormones, example testosterone. They could be able to cross the membrane barrier and control the gene expression. And example, secondary sexual character development and ovulation and spermatogenesis can be controlled by steroid hormones. And hydrophobic receptors are they able to travel the cell membrane from exterior to interior. Hydrophilic receptors will resides on the exterior surface and they interacting with the peptide hormones, example growth promoting hormones, epidermal growth factors, and acetylcholine receptor present on the neural cells. These all are the good examples of hydrophilic uh, ligand molecules, signal molecules. Then, what is the main mechanism of signal transduction? A signal molecule is a ligand information carrier will searching for its own receptor as like magnet and iron, enzyme and substrate interaction manner. The interaction between the ligand and receptor will mediate the cytoplasmic domain changes. A conformational change in the receptor domain will recruit the primary transduction of the signal molecule. Sometimes the cytoplasmic primary transduction domain will require many signal molecules called secondary messengers. The secondary messenger will relay the signal. The second mechanism is called relay, release of the signal into inside cytoplasmic side, where a modulation of signal can be takes place as like a regulator, fan regulator. The signal going to be modulated, excess signal or lower signal. If the signal is amplified many times, the multitude of a particular signal is called signal amplification. So minute ligand concentration will leads to huge signal accumulation is called amplification. The third stage is called amplification. After amplification, signal going to be divergence. This is targeting multiple targets. Cellular target, nuclear membrane, gene expression, and cellular activities. The three are controlled at a time. So this is called signal transduction. The way of signal delivery and amplification and control over the target is known as signal transduction. The signal could be different types of signals. The present exterior side will searching for their receptors. Receptors are a protein molecules present on cell membrane which will targeting the different types of ligand molecules for cell synthesis and cell control. So the receptors are vary in their nature. Now intracellular relay systems will leading to promoting signal cascade. A series of events of reactions will be takes place in the cell, one upon the other will be linked together to carry out a specific biological function. The intracellular targets may be cell synthesis or cell regulation mechanisms. So after particular cell signal cascade recruitment, different critical functions of system will be able to recruited and they lead to signal cascade. The components of signal cascade are very important. They are called signal transducer as like a plug. Signal transducer is a signal molecule information carrier. They carry the information from a chemical molecule to the its receptor. On receptor binding, it transducing the information downward, that is called a relay. Now it signal from point of reception to point of response or target will be released outside called relay, that is called signal relay. And signal relay then followed by signal amplification. Now signal can be amplified or regulated, low frequency, high frequency, and distribution of signal across the body or particular given system, and then modulation of the signal according to given relay system. Now the biological function could be modulated in fine tuning manner. This transducers, relay systems, amplifiers, distribution and modulation systems are also equivalents to radio tuning, TV relay systems. For example, in a radio, there is a signal, nothing but our information, which is recorded by a microphone. Microphone is connected to a device called transducer. From transducer, it will transducing the signal. So physical form of signal is converted into sound form. Sound form is converted into electrical form. Within the amplifier, now amplification will be taking place. We can control the signal high tune, low tune, high volume, low volume, then followed by uh, receiver. Receiver nothing but a TV set or a radio set that going to be receives the relay system. Now the amplification will be fine tuned. We can also control the volume key up and down, like that amplification takes place. The entire system is called controlling the signal level, high level, low level, high value, low value, is called modulation and distribution. So we can connect the signal to different types of devices, a distribution, like that. The biological system also obey the signal transductions in the